I'm going to be giving you an overview of the work we do in the lab. We use computational systems approaches to understand immunological disorders. More specifically, we use machine learning and network systems biology approaches to understand molecular mechanisms of natural and vaccine-mediated immunity. And you can think of research in the lab being divided into two broad arms. One is the use of machine learning approaches on multiomic data sets, transcriptomic, proteomic, metabolomic, antibodyomic, cellular phenotypic. So these large data sets, how do we systematically combine them to understand correlates and mechanisms of natural and vaccine mediated immunity? The other is how do we integrate these data sets taking into account prior knowledge biological networks such as the protein-protein interactome network or gene regulatory networks to understand mechanisms of natural and vaccine mediated immunity. And I'll just give you like a 30,000-foot view of ongoing research. So for example, in an antibody context, people may study antibodies in isolation, but we take this broad systems overview, systems immunology perspective to studying antibodies. So we think of each antibody as having a barcode. The barcode is determined by the subclass and glycosylation profile of the antibodies. And in a polyclonal pool of antigen-specific antibodies, you have many of these barcodes. So using machine learning approaches, we can interrogate what we call the antibody-ome, this entire spectrum of antibodies, and figure out what kind of properties of antibodies, functional or biophysical, are tied to disease outcomes or disease states. So the question we would ask is, are there specific humoral biomarkers that can distinguish between disease states? And then what do those biomarkers tell us about the actual underlying molecular mechanisms? So we've used such approaches in a wide range of contexts, including dissection of correlates of vaccine-induced immunity in HIV, differences between latent and active tuberculosis, correlates of protection induced by the malaria vaccine. And the point is not to overwhelm you with all of this data, but to just really give you an overview of the wide range of disease contexts we work across, taking a systems perspective. So the take-home message is you have many different multi-omic data sets. How can you, using principled approaches from machine learning and network systems biology, combine them to understand both biomarkers as well as molecular mechanisms underlying natural and vaccine mediated immunity across a range of immunological disorders? So possible rotation projects include using machine learning and network biology approaches to combine transcriptomic and epigenomic datasets to uncover modules that regulate the developmental trajectories of T follicular helper cells. Another possible rotation project is using these network approaches to decipher causal networks in rheumatoid arthritis. These are simply two examples, but there are several other related projects that I'm happy to talk to you about. And prior programming experience is a bonus, but definitely not a requirement. So please email me if you have questions or if you want to set up a rotation. Look forward to hearing from you. Thank you very much.